Everyone, so today we are going to be looking at non-Mendelian genetics. So far, everything we've been doing with genetics and Punnett squares has been Mendelian genetics. Um, so it follows the rules that Gregor Mendel proposed, that there are dominant and recessive alleles. Homozygous, heterozygous, dominant, recessive, all of those terms, right? If a plant is, uppercase G, little g, then the uppercase G is going to be the phenotype, right? So that is what Mendel proposed, and it is actually a miracle that he worked with pea plants because pea plants follow Mendelian genetics so perfectly. He happened to choose a plant that has all of those two-factor genes. The peas are either yellow or green. The peas are either wrinkled or smooth. The plants are either tall or short, and it follows those rules perfectly. But since Mendel discovered this and proposed these rules, scientists have discovered situations where Mendel's rules don't apply, and we are going to look at the four examples of that today: sex-linked inheritance, incomplete dominance, codominance, and multiple alleles. So the first one we're going to look at is called sex-linked inheritance. Sex-linked inheritance has to do with the X and Y chromosomes, the sex chromosomes. So if you remember back to our cell division unit, we learned about all the different types of chromosomes in a cell. Humans have 22 pairs of regular chromosomes. They're called autosomes. And then one pair of sex chromosomes. The sex chromosomes determine your sex. If you have two X chromosomes, you are a female. If you have a X and a Y, you are a male. So this picture is actually of an X and a Y. So this organism would be a male, okay? And when we talk about sex-linked inheritance, this is, the inheritance of genes on the sex chromosomes. So the X chromosome carries a certain set of genes. There's all different sorts of genes that are found only on the X chromosome. And then there are genes found only on the Y chromosome. For example, a very important gene on the Y chromosome is called the SRY gene. The SRY gene turns on during development if the developing embryo has a Y chromosome. The SRY gene will be present, and it will be turned on, and it will produce testosterone, which leads to the formation of the male sex organs. So, this is why having a Y chromosome makes you male. If you don't have the Y chromosome, then the testosterone never gets made, male sex organs never develop, and the default female organs are produced. So females are XX, males are XY. Now, let's say there are mutations or different alleles present on these chromosomes. The pattern of inheritance of X and Y chromosomes does not perfectly follow Mendel's laws. So we're going to take a look at this, okay? So like I said, it's associated with the X or Y chromosome. And because this has to do with sex, you'll see it usually only in males or females. It's more heavily weighted on one side or the other. And the notation we use is a little bit different. Instead of capital and lowercase letters, we use X's and Y's, okay? To show a mutated X chromosome, use X minus. To show a normal X chromosome, use X plus, okay? So this is an example of sex-linked inheritance. We don't know what gene is affected here, but we know it's on the X chromosome. And that's usually how it is. Usually these genes are on the X chromosome just because it's bigger. There are more genes on this chromosome. So this male has a good X. This female, this mother, has one good X and one bad X. The male doesn't have two X's, right? So he only has one copy, and it happens to be a normal copy. He then has a Y chromosome, which is irrelevant in this case because the Y chromosome has different genes. It doesn't have this gene. But the mother has a good copy and a bad copy, and because the bad copy is recessive, it is hidden in the phenotype. She's just a carrier for this gene. She doesn't have whatever disease would this mutation would cause, but she's still a carrier. She can pass this X chromosome on to her children. So you don't see the phenotype, but it's still in her genotype. So if you were to do the Punnett square for this cross, you'd see that an unaffected father would be X plus. These are the sperm he could make. He can make a sperm with his good X, or he can make a sperm with his Y chromosome. The mother is, remember, a carrier, so she can make an egg with her good X, or she can produce eggs with her bad X. And remember, that all comes down to crossing over an independent assortment during meiosis. So let's say, this egg and this sperm come together. You would have a girl because it's an X, and this would be an unaffected girl. Let's say this egg and this sperm come together. You would have a carrier female. Unaffected, she still has that dominant good copy, so you don't see it, you don't see the problem, but she's still a carrier. For their sons, so let's say this sperm that carries the Y combines with this egg, you have an unaffected son. This son would be affected. He only has one copy of X. He doesn't have the backup like the girl does. So if he gets this X from mom, he's doomed because he doesn't have the backup from dad. He's a boy. His dad gave him his Y, right? A male's X chromosome can only come from the mother because their Y chromosome came from dad. Dad can give them nothing else but Y because they're a male, okay? So a classic example of sex-linked inheritance, one of which, so this is a funny picture, but I guess hairy ears, the gene for ear hair is on the Y chromosome. So it's only ever seen in males. So if males have that gene given to them from their father, they will have hairy ears. You never see females with hairy ears. A gene on the X chromosome determines color blindness. So just a little bit of background. Color blindness does not mean you only see black and white. The most common type of color blindness is called deuteranopia. I don't know if I'm saying that right. But basically people with red-green color blindness can't tell the difference between red and green. It just looks like a yellowish brown, okay? So when they see pictures like this, right, if we go back to our slides, they don't see the distinction between the red and the green, so it just looks like yellowish green dots. People who are not colorblind can tell that this is 45, okay? So if you don't see a 45 in here, you are potentially colorblind. It is more common in males because it's on the X chromosome. Females, in order for a female to be colorblind, she has to have 2 bad X chromosomes, which is rare. For a male to be colorblind, he only needs 1 bad copy from mom. So that's easier to get. You only need 1 affected parent. You don't need both parents to be affected. So it's more common in males. Another example of sex-linked inheritance you're familiar with is adrenoleukodystrophy. This is Lorenzo's oil. Lorenzo, his disease, adrenoleukodystrophy, the mutation is on the X chromosome. So this is called a pedigree. Pedigrees can be used to trace a mutation through bloodlines, through family. So you can see here's Lorenzo. And then remember, Augusto, the father, had 2 kids from a previous marriage, Christina and Francesco, but neither of them were, they did not have adrenoleukodystrophy. So we know what their phenotype would be. We know what their genotype would be. Lorenzo has to be, they're not using the plus and minus. This would be X minus, right? X recessive and Y because he's a boy. And if we look at his parents, Michaela and Augusto, Michaela had to be a carrier. Augusto, he gave the Y. And we know that he did not have adrenal leukodystrophy, so he had to have a good copy. Michaela is right here. She is a carrier. She has a good copy. And that's what you see, right? She doesn't have adrenal leukodystrophy, but then her bad copy was passed on to Lorenzo. And then you also see her sisters. So Deidre was the sister most prominently shown in the movie. She had the short red hair. She was like the guinea pig that they used Lorenzo's oil on first to see if it would work. She was a carrier, so she had some mutated proteins, but her body, relied heavily on her dominant copy of the gene, kind of ignored her carrier copy. Oh wait, this is Deidre, sorry. So she was a carrier. So they were able to test her blood levels to see if Lorenzo's oil helped. And then these are Michaela's other sisters, all of which either were not carriers, or in this case, Knies was a carrier, but she only had girls, right? So the girls get the dominant copy from dad. So they are not going to have adrenal leukodystrophy. And you can trace it back to Michaela's mother, right? The mother is the one that passed on her bad X. If Mr. Murphy had adrenal leukodystrophy, he wouldn't have lived to the age of 7. He wouldn't have been having children. So, he would have never passed on a bad copy of X to begin with to Michaela. So Michaela only got her bad copy of X from her mother. And then Michaela, she's right here, sorry. The labeling's bad. This is Michaela, this is Augusto. Michaela had a 50-50 chance. She could have passed on her good X or her bad X. Even if she had passed on her bad X, if Augusto had passed on his X, they would have had a girl who was a carrier. But Augusto passed on his Y, Michaela passed on her bad X, and you get Lorenzo. This is actually their Punnett square, right? Here's Augusto, here's Michaela, and then this is Lorenzo. They had a 25% chance. They only had one child together. A 25% chance of having adrenal leukodystrophy. 75% chance of not having it. I just want to pull up the clip from the movie where they talk about this. It probably went over your heads back then, but now you'll be able to see it with a little bit of clarity.
Spinach and peanut butter make the difference.
The human body needs these fats in order to create cells. And the excess is burned off. That's normally, of course. But in the case of an ALD boy, these saturates can't be broken down, and they build up in the brain.
I mean, somehow this buildup strips the myelin covering.
Yes, exactly. And for the moment, we don't know why this is, but we feel that by withdrawing the saturated fats from the diet, we will prevent them accumulating in the brain.
And if we could?
There's no way to reverse neurological damage. All we can hope for is to slow the cascade of symptoms. But if it's any consolation to you, you'll be helping us understand the biochemistry of this heartless disease. So, can we enroll Lorenzo in our trial?
I suppose so.
Good. Then I will pass you over to our dietitian. And meanwhile, we should arrange some genetic counseling.
Oh, Dr. Nicholas, it's highly unlikely that we will have another child.
Oh, no, but your sisters and their daughters, they may all be carriers. We need to test them.
Only my family? What about his?
No. ALD is passed only through the mother. But I thought you understood all this.
We knew it was inborn, but we assumed that meant a combination of our genes.
That's true in other inherited diseases, but ALD is only carried on the female chromosome.
Excuse me. Are you saying that Lorenzo got this directly from me?
Well, in the sense that ALD is sex-linked, yes. It goes from mother to son.
Then how did I get it?
Well, the woman gets it from her mother.
And if I inherited the defect, why don't I have the disease?
No, the woman is only the carrier, nothing else. But with each conception, she has a 50-50 chance of passing on the defect. And when that happens, it's the cruelest kind of genetic lottery.
Lottery.
Yes, it's a clumsy word. I use it because it's...
So you get the idea. It's a terrible circumstance that, she really did have a 50-50 chance of passing on her defected X chromosome. It's partly Augusto's fault too for passing on the Y and making a boy when he could have passed on his good X, but it's what happened. So that's another reason why throughout the movie they say ALD boys. In order for a girl to have ALD, she'd have to get a bad X from her mother and a bad X from her father. But if her father had a bad X, that means he has ALD. So he wouldn't have lived past the age of 7, which means he wasn't having kids. So that's why it's only seen in boys. Another example of an X-linked trait is male pattern baldness. Again, it's on the X chromosome, so it's more often seen in males. Yes, women can go bald, but it is more often in men. So if you don't know who these people are, this is Prince Charles, the son of the Queen of England. This was his wife, Princess Diana. She died in 1997 in a car accident. And they had 2 kids, William and Harry. You might know Harry. He's married to Meghan Markle, just had a baby, Archie. And then this is William, who got married to Kate Middleton. But, anyway, it is well known that William once had a full head of hair and he now does not. And Harry is starting to go bald. So, obviously it would make sense to blame their baldness on Charles because he's balding. But because it is on the X chromosome, you know that Charles gave a Y. They had boys, right? So the fact that they are balding has no relation to the fact that Charles is balding. Charles is balding because of the Queen. William and Harry are balding because of Diana. Now, Diana had a full head of hair. You wouldn't think of it, but she was a carrier. She had one good copy of X, one bad copy of the balding gene, right? And when she had William and Harry, when they were conceived, she passed on her bad X in both cases. Okay? So, gentlemen, listening in, if you are concerned about one day losing your hair, do not look to your father. Look to your mother. If your mother is balding, you are doomed, right? That means that she has only bad X chromosomes and only gave you a bad X chromosome. If your mother has a full head of hair, you have to look at her parents. If her parents are balding, then there is a good chance that they passed on that X chromosome to her, and then she could have passed on that X chromosome to you. So, there you go. Ladies, if you're wondering about going bald, you would have to look at both of your parents, right? Your mother would either have to be balding or be a carrier, and your father would have to be balding, pass on. You would have to have both bad copies of X, right? Males, you only need the one bad copy of X from your mother. So, a practice problem. A colorblind man marries a female who is a carrier for color blindness. What is the probability that they will have a child who is colorblind? So the hardest part of these non-Mendelian practice problems is getting the notation right, I think, getting the problem set up. Once you get the Punnett square set up, you're fine. So a colorblind male, you know that a male has to be XY. And you know that mom has to be XX. Okay? Remember that you don't use the capital letter, lowercase letter in this case. Use X and Y. The father is colorblind, so he would be X minus. The female is a carrier, so she is plus and minus. So then you just fill in the Punnett square. Now, in this case, colorblindness isn't deadly. So, this father, being colorblind, can pass on his X minus to a daughter. And if the mother is a carrier, then you have a colorblind female. It's rare, but this is possible. And then here's your colorblind male, right? Only inherited the trait from mom because the Y is a completely different chromosome, doesn't carry that gene, and so he only has that X chromosome to use. So what is the chance that they will have a child who is colorblind? It is 50%. 2 out of the 4 are colorblind. Okay? So moving on, our next type of Mendelian genetics is incomplete dominance. So this is when you have two alleles that are both not dominant, right? They're both not all the way dominant. So what we've been looking at so far is where one allele is dominant, one allele is recessive, right? Brown eyes, capital B, is dominant to blue eyes, lowercase b. If you are heterozygous, you have big B, little b, you have brown eyes because big B is dominant, right? That's what we've learned so far. There are cases in nature, however, where neither allele is completely dominant. So a perfect example is in the flower known as a snapdragon. Snapdragons can be red, white, or pink. So that means that red and white, the alleles for flower color, both red and white are, or I should say neither red and white are completely dominant. When the flower is heterozygous, when they have one red allele and one white allele, one doesn't outcompete the other. Instead, you get a blending of the two. So you get pink flowers. This notation is a little bit different. Again, you don't have uppercase, lowercase. You have all uppercase. They're all like sort of dominant. And it's written with numbers. So in this case, red flowers are R1, white flowers are R2, and then the heterozygous R1, R2 would be your pink. So what happens if you cross two incomplete dominant flowers, two pink flowers? You would have R1, R2, R1, R2. Then you do the Punnett square. This would be R1, R1. That's red. R2, R2 is white. And then these flowers are your heterozygous. Neither red or white is dominant, so you get a blending of both, which is pink. Another good example of incomplete dominance is hair texture. So this mother has wavy. They both have wavy hair. So this mother can make eggs that are curly or straight because she is heterozygous. She carries both genes. Father, same thing. He can make sperm with the curly allele or carrying the straight allele. So when they have children, if this egg meets this sperm, then you're going to get curly hair, C1, C1. These two are heterozygous. They would be wavy, and then C2, C2 is straight. It doesn't work that perfectly. There are some nuances in the genetics, but it's sort of the idea. Okay? So a practice problem. A red carnation is crossed with a white carnation, and the resulting offspring are all pink. What would be the result of a cross between a pink and a white carnation? So, again, you have to think of the notation. We're using all uppercase letters here because it's incomplete dominance. There's no recessive allele. There's no lowercase. So I'm going to choose, I'll choose R again. It can be any uppercase letter. You just want to make it letters, in this case, it actually doesn't matter if they look different, uppercase, lowercase. You know what, in that case, I'll pick any random letter, so I'll do a P. Pink flower would be the heterozygous, right? Because red and white make pink. So you'd have a heterozygous parent, P1, P2, both red and white, and then a white carnation will make P2. Okay, and then that would mean that red is P1. So then you do your cross, P1, P2, P1, P2, 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 P2. Okay? So what would be the result of a cross between a pink and a white carnation? So it's not asking you anything specific. It's not asking you for any percentages or anything. So this is your answer. These are the potential results, right? If you crossed this flower with this flower, these are the 4 potential offspring. Half would be pink, half would be white. Okay? So again, probably the hardest part is setting up the Punnett square, setting up the parents. And then once you go from there, it's pretty straightforward. Okay, next one is called codominance. Codominance is slightly different from incomplete dominance, but they're very similar. In this case, both alleles are equally dominant. Okay, so you actually see both of them at the same time. They're not competing with each other. They are equally dominant, right? Well, actually, I should say that incomplete dominant, they weren't competing for each other. I guess in codominant, they are, right? They're both trying to be seen. So in this case, the notation is 2 different uppercase letters, right? There's 2 dominant alleles. It's not one uppercase, one lowercase. Literally, they're both dominant. So you see both at the same time. Now, the difference is you don't see a blend. You literally see both of them at the same time. So in chickens, this chicken would be big B, big B. This chicken would be big W, big W. And then when they have offspring, it's a big B, big W, and you have a checkered chicken, both black and white feathers, not gray feathers. That would be incomplete dominance. You see both at the same time, okay? Another example is the rowan coat in cows and horses as well. So if you cross a pure red with a pure white, right here, pure red with pure white, you get rowan cows. They have splotches of both red and white, okay? So they are co-dominant. So an important distinction, incomplete dominance is when you see a blend of both, right? They mix together, red and white make pink. In codominance, you see both red and white. So practice problem, in cows, red and white coloration alleles are codominant. A cow that has both alleles would be roan. A roan cow is crossed with a red cow. So we have a roan cow. Remember, the notation here is 2 different uppercase letters. So a roan cow would be RW. They give you the notation up there. With a red cow, RR. So RR, RR, RW, RW. And then the question, what percent of the offspring will be red? These are the red ones, 50%. These would be roan. Okay? So now our last example. It's called multiple alleles. Okay? So far, everything we've looked at has had 2 alleles, right? Tall, short, green or yellow, curly or straight, round seed, wrinkled seed. There's only ever been 2 options, but we know that there are things that exist with multiple alleles, one of which is hair color, right? There's not just brunette and blonde. There's red, there's black, there's all sorts of variations, right? So this is where multiple alleles come in. Probably the most common example, the most universal example, are the human blood types. So there are actually 4 different blood types, but there are 3 different alleles. A, B, or O. Okay? So this is, it can have any, it's not only when there are 3 alleles, it's when there are 3 or more. It's anything more than 2. Blood typing is the most common and accessible example. So this is how blood typing in humans works. Let's say you have So let me back up a little. So everyone is red. Sorry, everyone has red blood cells, but what's different about those red blood cells is what sticks out of them. So if you remember, cells have that cell membrane, and things stick out of that cell membrane. There are transmembrane proteins, peripheral proteins, blah blah blah. On the surface of all of your cells, you have these things called antigens. Antigens are proteins that stick up out of the cell as kind of like an identification tag. It's kind of like a name tag. These antigens identify this red blood cell as your red blood cell. Okay? So your white blood cells know not to attack this red blood cell because it has your antigens on it. So if you are type A, then you have type A antigens. If you're type B, you have different antigens. You have type B antigens. And your white blood cells know not to attack and kill this red blood cell because it has these identification tags. It has B antigens. However, if you are type A, you have antibodies that are designed to attack B antigens. Let's say you are type A and you are supposed to have these red blood cells floating around, but for some reason you get a transfusion or you drink blood or I don't know, whatever the case may be, and you have these red blood cells floating around in your bloodstream. These would be recognized as foreign because it has the B antigen, not your A antigen. So you have antibodies that are designed to attack and kill anything with the B antigen. Okay? So because of that, if you are type A, you cannot have B or AB blood in your bloodstream because you have antibodies designed to attack it. So type A blood has type B antibodies, whereas type B has type A antibodies, and therefore, if you need a blood donation, you cannot accept blood from anyone who is type A or AB because you have antibodies that attack those cells. The plus and the minus is something else entirely different. It's called the Rh factor. So don't get into that, but the AB, all that is what we're talking about here. Now, if you have type AB blood, that means that you have both the AB antigens. Now, this is interesting. This means that let me back up. If this red blood cell, if you're seeing both the A and B antigens, what does that mean? Is the AB blood type an example of codominance or incomplete dominance? The answer is codominance. Okay, you're seeing both A and B. So people with AB blood have no antibodies, right? If they had anti-B antibodies, then your own body would be attacking your blood cells. Okay, so you don't have any antibodies, meaning you can actually accept any type of blood. You have no antibodies to attack red blood cells. So you are what's called the universal acceptor, the universal recipient. The most common blood type is type O, right? You have no antigens. You have nothing on your red blood cells that mark it as being foreign. This is evolutionarily advantageous because you can accept type O from anyone. Or I'm sorry, no. Wait. I got that backwards. You have all of the antibodies. So if any of these enter your bloodstream from a foreign invader, your body is designed to attack it. So that is why it's evolutionarily advantageous, right? You can have all the antibodies. You have the best protection system, right? If any of these cells get into your bloodstream, they are going to be attacked. You have no antigens, all the antibodies. So type O can only get blood from other type O people. You cannot have any antigens on your red blood cells or it'll be attacked because you have all the antibodies. But you can be the universal donor. You can donate blood to any of these people because there are no antigens on your blood to, raise alarm from the antibodies. So that's how blood typing works if you're interested. Now let's look at how the genes actually line up. So let's say one parent has an A allele. Now look at the notation of this. For some reason, they use the letter I. I'm assuming it's because antigens and antibodies are all often called immunoglobulins. But anyway, remember that it is the I. Okay? I capital A is type A blood, while I capital B is type B blood. Lowercase I, we know that lowercase means recessive, and that is type O. So let's say, it doesn't even matter if they're male or female. This parent gives I capital A, this parent gives I capital A. The offspring's genotype is homozygous A, and the blood type is A. If one parent gives an A, the other parent gives a B, the offspring is type A and type B, right? Because both of these are capital letters and they're codominant. So right there, you have an example of codominance. Their red blood cells will have both A and B antigens. One parent gives an A, one parent gives an O. The O is recessive, and this person's blood type will be A. The O disappears, right? It's still in their genotype, but they still carry that. They can pass it on to their kids, but their actual blood type, is determined by this dominant allele. All of their red blood cells will have A antigens on them. And then the only way you are type O is if you are homozygous recessive. Little I gives you O, and it's actually the most common blood type, okay? So blood typing is unique because it's an example of multiple alleles, right? There's IA, IB, and little I, but then it's also an example of codominance as well. So let's do a practice problem. A father has type AB blood and the mother has type O blood. What percentage of their offspring would you expect to have all the different blood types? So remember, the notation is a little bit different. You don't use A, B, and O. You use I's. So the father would be I A, I B, right? Because he's AB. Then the mother's type O. Type O is homozygous recessive, so she would be little I, little I. So then all the offspring would be I A, I A, I B, I B, I. These are terrible B's. So how many have type A? 50%. Remember, the I, unless it's homozygous recessive, the I goes away. So this person, this offspring, is type A, as well as this one. So 50% here. How many are type B? These two would be type B, 50. Are any of them AB? No, 0. Are any type O? No, 0. Okay? So again, probably the hardest part is just setting up the parents, figuring out what their genotypes are, and then from there you can figure that out. So now that we've gone through all of the different types of non-Mendelian genetics, you're going to go do your practice problems. If you need to come back to this video to see how I set up each Punnett square, feel free. Again, the hardest part is just setting up the parents. From there, getting the Punnett square is simple. And then always remember to look for these other questions, right? The question doesn't stop at make the Punnett square, right? It then asks you a little bit further, what are the percentages of all the different what are the percentages of all the different blood types? Okay? So you want to make sure you do a Punnett square for each question and then answer all of the little embedded questions within the larger question, if that makes sense. All right, so good luck. If you have any questions, please reach out. Hope you found this educational. This is probably going to be my last recorded lesson for the year. Next week we have all half days and we're going to be doing a little bit of ecology, just to tie everything together from the year. I've gotten some questions about final exams. I'll just say this while I have everyone's attention. There is no final exam. So once we finish next week, the 2 lessons on ecology next week during the half days, we are done for the year. So I hope you enjoyed your time with remote learning. I know it wasn't ideal. We did the best we could. Hopefully you still learned something. All of these topics, although there will not be a final exam, everything we've covered in remote learning is important and you'll need it for future biology classes. But luckily, if you ever need to go back and relearn any of this, you have all of the resources in Google Classroom. So if I don't hear or speak to you again until the end of the year, I hope you enjoy your summer. And hopefully we're back in September. Please come say hello. And I miss you all. Enjoy your upcoming summer. All right, bye.